Well, thank you. And I wanted to thank Hunter's Hope for the invite and allowing us as a company to present and provide an overview of Mirum and a little bit more about our culture and what we do and our focus and our commitment to the rare disease community. And also to allow us to present some of our data for Restore looking at CDCA or chenodeoxycholic acid in CTX patients. So let's begin by looking at our people, our commitment and culture at Mirum. And so when I think of Mirum, I think of patient centricity, and patient centricity is essentially the cornerstone of what we do and why we do it. This is our essential why. We're a small organization. We have about 250 individuals globally working for Mirum. We're right on the cusp of about 300 individuals, and our passion lies within the rare disease arena. So essentially, we're focused and committed to the patients, their families, caregivers, as well as the community we serve, first and foremost. And it's our passion and for what we do, and essentially that's what drives our need to make a difference and move the needle within the rare disease space. And so, we strive to be great partners and collaborators with unified purpose. And we want to be able to ensure that patients, globally are introduced to important new therapies that address unmet needs within the rare disease arena. So what we see here is the one thing I've seen grow essentially is our collaboration and support for patient advocacy groups and patient organizations. And what you see here on this slide is essentially our connection to the global rare disease community. And I'm very proud of the number of organizations we collaborate with, as well as the number of patients we touch. So a little bit more in terms of the growth opportunities and Mirum pipeline. There are three approved rare disease products, three additional indications in development and high need orphan indications. So we have Livmarli, which is both FDA and EMA approved for Alagille as well as PFIC. We have Chenodal or chenodeoxycholic acid for CTX. It's approved for radiolucent gallstones. It's essentially used primarily off label to treat CTX. And what I'll be presenting today is the Restore phase 3 study, which was kind of a catalyst to the submission of our NDA late last month for CTX. And then we have an FDA-approved product for cholic acid, which is the other bile acid for bile acid synthesis disorders, as well as PBDs and ZSDs. And then we have Volixibat, which is undergoing clinical studies. We've had positive interim analysis for both primary PSC as well as PBC. So a little bit more now on kind of the topic that I'll be discussing today, which is cerebrotendinous xanthomatosis or CTX. Now, CTX is essentially a rare progressive genetic lipid storage disorder. It's a metabolic disease. It's autosomal recessive in nature. And the mutation that occurs on the long arm of chromosome 2. And so essentially, when you look at CTX patients, they lack or have a deficiency or a defect in one of the enzymes that basically converts cholesterol to the two primary endogenous bile acids, both cholic acid as well as chenodeoxycholic acid. And so these individuals lack the enzyme known as sterol 27 hydroxylase, which encodes for CYP27A1. And that leads to drastic limitations in the primary bile acids and an increase in not only bile alcohols, but also cholestanol, which is a byproduct of cholesterol metabolism. So that cholestanol essentially accumulates or hyperaccumulates in various parts of the body, not only the lens of the eyes, but also the CNS, the cardiovascular system, as well as different tissues within the body itself. Now, when we look at this slide, we see kind of a timeline in terms of the most common cardinal features, the hallmark symptoms that are associated with CTX. So you see that we have chronic diarrhea, which can occur as early as birth and can continue throughout an individual's life. We have bilateral cataracts, that's idiopathic in nature. We have tendon xanthomas, as well as progressive neurological deterioration, which happens to be one of the most debilitating aspects of the disease itself. And so based on the fact that the disease is essentially very heterogeneous in nature, it leads to the variability in terms of onset and severity of the disease, leading to not only misdiagnosis and underdiagnosis, but also delayed diagnosis in these individuals. And we can see that based on the mean age of diagnosis here. On this slide, we see that it's about 36 years of age, plus minus about 12 years. And simply put, that mean age of diagnosis is just, it's unacceptable. And so what we want to be able to do is increase diagnosing CTX as early as possible, and basically arrest the natural course of progression of the disease, preventing that progressive neurological deterioration with positive clinical outcomes. So a little bit more on chenodeoxycholic acid, or CDCA. Now, CDCA was essentially approved by the FDA in 1983 for the treatment of radiolucent gallstones. And CDCA is essentially, it's considered the standard of care for the last 40 years or so, for the treatment of CTX, which basically decreases not only bile acid synthesis, but reduces cholestanol and prevents disease progression in CTX patients. Now, CDCA, as I mentioned, is not approved for the treatment of CTX, but it's essentially considered a medical necessity by both the FDA as well as the NIH, and we have ODD for CDCA as well in this country. Now, Mirum basically has both bile acids, both chenodeoxycholic acid, which is chenodol, as well as cholic acid, which is Cholbam, and it's available through Mirum and our Mirum Access Plus program. And through our discussions with the FDA, what we did was basically come up with a phase 3 study that would basically, provide the data needed in order to approve CDCA specifically for the treatment of CTX. So the study itself and essentially explores efficacy and safety of CDCA in patients with CTX. It's called RESTORE. It's a phase 3. It's a randomized withdrawal, double-blinded, placebo-controlled, 2-period, 2-treatment crossover study with rescue medication. Now, the study itself is a 24-week study. There are 2 open-label periods, period 1 and 3, and then the 2 double-blinded periods, period 2 and 4. Each of the open-label periods are 8 weeks in duration, so a total of 16 weeks for the open-label period. And each of the double-blinded periods are 4 weeks in duration, totaling 8 weeks. So total of 24 weeks, and at the end of the 24 weeks, there's an end of study phone call in order to assess safety. Now, the investigational product here, where all individuals basically receive that investigational product, is CDCA, or chenodeoxycholic acid, at 250 milligrams TID, which translates to 750 milligrams daily. Now, as long as these individuals met the eligibility criterion, which essentially clinical diagnosis of CTX and with biochemical confirmation, so increases in bile alcohol as well as cholestanol in urine or in plasma or urine, as well as these individuals being at least 16 years of age at the time of consent, as well as those individuals who are of childbearing potential using reliable contraception. These individuals were essentially given 8 weeks open label of CDCA. Now, there's 2 reasons why this happened. One is that we wanted to basically establish reliable baselines for all of the biomarkers that we're evaluating. And then also we want to be able to establish baseline clinical signs and symptoms for each of these patients because of the heterogeneity in this disease. So each individual would essentially be their own placebo or control. So after 8 weeks of treatment with CDCA, these individuals, as long as they didn't have any type of adverse events or intolerance to the drug itself, or didn't have any additional clinical signs or symptoms or worsening of those clinical signs and symptoms, these individuals are randomized to one of the 2 treatment arms for 4 weeks, whether either CDCA or placebo. After the 4 weeks, these individuals were returned to an open label for 8 weeks. And then after the 8 weeks of open label, these individuals were returned to the second double-blinded period, where these individuals who received CDCA in the first double-blinded period now receive placebo, and vice versa. Those individuals who received placebo in the first double-blind period now receive CDCA. Now, there are 2 rescue arms that were associated with these double-blinded periods. One was a biochemical rescue, and the second was a clinical rescue. So biochemical rescue are for those individuals who had the primary endpoint being 23S pentol, tenfold increase in 23S pentol from baseline. They were biochemically rescued. And then you had a clinical rescue in these double-blinded periods where you had, these individuals rescued if they had any type of clinical worsening of their clinical signs and symptoms. And here are the primary endpoints. We have the primary endpoint being 23S pentol or urine 23S pentol. We have the secondary endpoints here, 7-alpha-C4, 7-alpha-12-alpha-C4, cholestanol, as well as the percentage of placebo patients requiring rescue chenodeoxycholic acid during double-blind withdrawal periods, safety, as well as the exploratory endpoint, which is change in plasma bile alcohols. So in terms of the results, and just a little bit on the key demographics here, that age at study start was about 39 years of age. There's a greater preponderance of male Caucasians in this study and non-Hispanic or Latino individuals. These individuals were biochemically confirmed, but you can see that 100% of these individuals were also confirmed genetically to have CTX. And the mean age of CTX, age of diagnosis for CTX in these patient population was essentially about 35 years of age, plus minus about 12 years. In terms of most common CTX medical history in these individuals, I've just listed a lot, but the main three are essentially learning disability, abnormal gait or balance, and cognitive decline of any kind. But you can also note that based on the cardinal features of the disease, these individuals had, a good percentage of these individuals had xanthomas as well. And if you look at idiopathic diarrhea as well, or unexplained chronic diarrhea, there are 50% of these individuals who have that as well. So looking at the study itself and looking at the primary endpoint, urine 23S pentol, and what we see is this beautiful graph that basically represents the rapidly increased levels of 23S pentol after withdrawal of CDCA and the decrease of 23S pentol after restarting CDCA here. And essentially, this is a statistically significant 20-fold increase in 23S pentol at the end of each of these double-blinded periods compared to that of with CDCA. And if you look at the natural log transform scale, that translates to greater than 2,000% increase in 23S pentol during the double-blinded periods. And this also holds true in terms of the common theme. You see a statistically significant 2.8-fold increase in plasma cholestanol during the double-blinded periods versus that of CDCA. That translates on a natural log transform scale of approximately 177% increase in cholestanol. For 7-alpha-C4, there was statistically significant at a 50-fold increase of 7-alpha-C4, which was observed during the double-blinded periods compared to that with CDCA. And on a natural log transform scale, that translates to 3,900% increase in 7-alpha-C4 once these individuals were withdrawn from CDCA. And for 7-alpha-12-alpha-C4, it was statistically significant at a 14.3-fold increase on a natural log transform scale that translates to approximately greater than 1,200% increase in 7-alpha-12-alpha-C4. And finally, looking at plasma bile alcohols, it was statistically significant increase of 12.5-fold increase of bile alcohols here during the double-blinded withdrawal periods, and that translates to greater than 1,000% increase on a natural log transform scale. Now, when we look at the participants that require rescue medication during the double-blinded periods, what we see is that during the double-blinded period, there were 2 out of 7 individuals who required rescue. And then in the second double-blinded period, all individuals, 6 out of 6, required rescue with CDCA, which translates to about 62% of all individuals or participants who require rescue medication during the double-blinded periods. When we look at the common adverse events, the most common adverse events while receiving CDCA was essentially diarrhea, constipation, as well as headache. And the most, and the majority of adverse events were essentially mild to moderate in nature. It was transient in nature as well and considered not related to study medication. There was one severe a treatment emergent adverse event, a drug overdose in one individual that led to discontinuation of the study medication, and no deaths were reported in these individuals. So in conclusion, RESTORE is a first randomized double-blind placebo-controlled trial evaluating CDCA withdrawal in patients who were clinically diagnosed with CTX with biochemical and genetic confirmation. Now, the study itself shows that withdrawal of CDCA treatment rapidly resulted in significant increase in the biomarkers that we evaluated in this study, and conversely, CDCA treatment also led to increases in CTX biomarkers in these individuals in the double-blinded periods, which basically goes to show that it's consistent with the decrease in CYP27A1 induction and control of aberrant bile acid synthesis. And so a statistically significant portion of these individuals receive rescue medication or blinded rescue medication during the double-blinded withdrawal periods, and the CDCA was well tolerated with most adverse events that are self-limiting, mild to moderate in nature, and transient in nature, with no abnormal liver function tests observed. So the authors would basically like to thank each of the clinical participants and the families and investigators for their participation in this study. And we've basically, here are some contacts at Mirum. Please feel free to kind of reach out to us anytime you want. We have myself, we have Erin Murphy, who's an executive director for corporate affairs, and then Alina Barton, who's the senior manager for global patient advocacy. With that, I'll take any questions that you might have.